Hey everyone, let's take a look at Mendel's, modified Mendelian ratios. To start, let's just briefly review Mendel's standard ratios, right? The monohybrid ratio, genotypic ratio of 1:1 in the F2 generation. Phenotypic ratio is 3:1, and genotypic ratio of the dihybrid cross, right? It's this long one right here, dihybrid cross in the F2 generation. That's the genotypic ratio, and the phenotypic ratio of the same generation is 9:3:3:1. So I think Mendel's work, I believe it was published in the 1870s, and it was about 30 years before his work was rediscovered by scientists who were studying chromosomes. They were finally able to track chromosomes during meiosis, and they realized, oh wow, there's this guy, the chromosomes are behaving like this monk, predicted they would. So Mendel's work had been rediscovered, and it was very exciting. And we had some explanations for the basis of how heredity was transmitted from parents to offspring. But interestingly, scientists, once they confirmed Mendel's results, they started to get more excited by examples of genetic crosses that did not produce Mendelian ratios. So we would call these non-standard ratios, or simply not non but modified Mendelian ratios. And the reason why we would use this one right here, modified Mendelian ratios, even though it means the same thing, is that the new ratios weren't the result of Mendel's postulates being inaccurate. They were the result of genes that were unlike the genes Mendel had studied, for example, or phenomena that were not observed by Mendel, but Mendel's postulates still explain transmission of genes between parents and offspring correctly. So what are some of the phenomena that lead to modified Mendelian ratios? So linkage, so that would be genes close together on the same chromosome. Mendel did not study genes that were linked. He did not study genes that were on sex chromosomes. So that can also lead to modified ratio. He did not study alleles that demonstrated incomplete dominance over an alternate allele, right? He did not study codominant alleles. And he didn't study genes that were involved in a gene interaction. And we'll talk more about what gene interaction is later on in the video, or in a subsequent video. So all of these conditions here, and probably some others, can lead to what we would call a modified Mendelian ratio. So now might be a good time to talk a little bit more about alleles. So as I have said before, alleles are different versions of the same gene. And as we'll talk about more in the future, and as I am sure you have learned in previous biology courses or in high school, DNA sequence, or genes have a DNA sequence, right? Genes have a DNA sequence, which is a specific order of A, G, C, and T nucleotides. So two alleles may differ by as few as or only by only one nucleotide. So they may be almost identical. But if it differs by one nucleotide, well, we consider those two different alleles. Or two alleles may differ by hundreds, thousands of nucleotides. The longest gene in humans is about 1 million bases long. And that's the dystrophin gene. So you could have 1,000 nucleotides different in there between different alleles for the same gene. In diploid species like humans, at most, one individual can have at most 2 alleles in one individual. But at the population level, and we can consider population to be all the potentially interbreeding members of a species, so we can assume all the humans on Earth are considered one population. So there could be thousands of alleles in the human population for a single gene. But at most, each of us only has 2 alleles for each gene. Now, if we can have all these alleles, which allele is the normal one? Well, the most common allele in a population is what we would call the wild type allele. And typically the wild type allele is associated with, say, the normal or the most common phenotype of the species. So the wild type allele, the normal allele, would be the one that is the most common in the species. And we are going to have a whole section, maybe a whole lecture or two, devoted to mutant alleles. But we should talk about mutant alleles a little bit before we start talking about modified Mendelian ratios. So let's say the wild type, which I'll abbreviate often as WT, wild type allele is normal. And let's call everything that's not wild type, we'll call it a mutant allele. So there are several different types or ways to classify a mutant allele. We can have a gain of function allele. We can have a loss of function allele, null allele. We can also have a neutral allele or neutral mutant. This little arrow right here saying I should put mutant in there. Neutral mutant allele. And with a gain of function allele, that would be an example of let's say if the gene is for a protein coding gene, gain of function would be increased function or new function. Loss of function would be decreased function of, let's say, the protein encoded by the allele, right? Some genes, lots of genes, the purpose is to make a protein or encode the instructions for making the protein. So a loss of function allele would lead to decreased function of that protein. The null allele would say eliminates function. Completely non-functional protein is the result of a null allele. A neutral allele would be a mutation that has no effect on function. And we'll see how certain mutations can have no effect on function of a protein. So how is it different from the wild type allele? It just has a different DNA sequence, but it has no effect on function of the protein. Let's have an example here. We'll see a lot of this moving forward in the next couple of videos. So let's say we have a gene that is involved in pigment production. It could be a pigment for body color, fruit color, fur color, some sort of pigment that adds color to an organism. So let's say there is some white precursor chemical or white pigment. Now, the gene could make an enzyme that converts or encodes instructions for making the enzyme that converts the white precursor into a colored pigment. So we'll say there's an enzyme here. Now a gain of function would lead to more color. So that's an example of a gain of function allele. See, loss of function, less color. Null, no color. And neutral, a neutral mutant allele, no change from wild type. Same color. So let's say the color was red. This one, or let's go with blue. More color would be dark blue. Less color would be light blue. No color would be white. Same color would be blue. So this is just an example of what these different types of mutations, the effect they could have on the biology of an organism. Okay, so in our next video, we will take a look at incomplete dominance.